with you, and I'm reading this because
That's
I
fine.
memorize it all, but welcome everyone, and hopefully there'll be some people online too, but it's my pleasure to introduce Erin Mordecai. She's an associate professor of biology at Stanford University, and she did her BS at the University of Georgia and then a PhD degree at UC Santa Barbara in ecology, evolution, and marine biology. Like multiple directions all at once,
Yeah.
which is exciting. And she then completed a 2-year NSF postdoctoral research fellowship in the intersection of biology and mathematical physical sciences and engineering at University of North Carolina at Chapel Hill and North Carolina State University. She's been at Stanford since January 2015. And her research focuses on the ecology of infectious diseases, including how climate, species interactions, and global change drive infectious disease dynamics of humans and natural ecosystems.
Great.
Thank you. Welcome.
Thank you. Thanks for having me. Thanks for coming, everyone. I'm excited to be here today at BU and see some new faces and some familiar faces as well. Today, I want to share some research that we've been doing in my lab over the last 5 or 10 years, studying the interactive effects of climate and land use change on vector-borne disease dynamics. Before I jump into the research, I want to take a moment to acknowledge the funding sources that have made this work possible, including NSF, NIH, and several of the Stanford institutes and centers, as well as some of our important collaborators listed over here on the side that have worked with this research with my lab. And more specifically, I want to acknowledge a large group of people from my own lab, past and present, that have really been the drivers of this research. And I'm really proud to call these people part of my lab and to have gotten to work with them. So today's talk is going to focus on vector-borne disease. And in case you're not familiar with what that term means, vector-borne diseases are diseases that are caused by pathogens that are transmitted by biting arthropods, like mosquitoes, ticks, flies, or fleas. And the reason that we focus on these diseases is really twofold. One is that they're some of the most important infectious diseases that impact humanity. These include diseases like malaria, dengue, yellow fever, West Nile, Lyme disease. So a lot of very high burden diseases and a lot of diseases that are increasing in burden. The other reason to focus on them is a more ecological reason, which is that these diseases rely on some sort of arthropod vector for transmission, and arthropod vectors depend on aspects of their environment, like habitat and climate. So vector-borne diseases really form this kind of lever by which human impacts on the environment feed back to affect our health. So we're interested in vector-borne diseases from this double perspective of both the public health burden, but also their ecological and evolutionary dynamics. And vector-borne diseases cause a large burden, as I alluded to earlier. We estimate that there's somewhere between 58 and 96 million symptomatic cases of dengue every year. So a very large burden of a very severe febrile illness that it doesn't lead to a lot of deaths, although it does cause some deaths as well. And malaria, of course, causes a really large burden, predominantly in women and children, or pregnant women and children in sub-Saharan Africa. And currently, there are about 619,000 deaths every year from malaria, despite really massive efforts to try to control these parasites. So these are just two examples of the high burden of vector-borne diseases and the reason that we really care about them from a public health perspective. From an ecological perspective, these diseases are changing really rapidly because of human impacts on the environment. Of course, we're all familiar with the impacts that humans are having on the climate through our carbon emissions, but we're also really rapidly modifying landscapes. We're converting land from natural ecosystems into urban areas and agricultural areas. We're moving people and goods and species around the world at unprecedented rates. And we're having really pervasive impacts on almost all of the Earth's ecosystems. So the question my research program is focused on is how is this global change altering the dynamics of infectious disease, and particularly vector-borne diseases? This is a complex question to address because vector-borne diseases, like most other pathogens, are embedded in complex ecological systems. So this is just kind of a cartoon diagram representing this idea. A lot of vector-borne diseases and other infectious diseases rely on interactions between multiple species of hosts and vectors, represented by those little arrows. Often the parasites and pathogens that cause these diseases are interacting with other parasites and pathogens, both within the same host individual and within the same host populations. And both the parasite communities and the host and vector communities are affected by multiple aspects of the environment. So anthropogenic change, or human-driven environmental change, is expected to have multifaceted impacts on infectious disease systems. And that leads to two particular challenges. One is complexity. So the fact that there's lots of potential ways that this system could respond to environmental change. And the second is nonlinearity. What I mean by nonlinearity here is that a simple incremental change in an environmental variable doesn't necessarily result in a simple incremental response in the disease system. And that can make it really difficult to predict how infectious disease systems will respond to human-driven impacts on the environment. So because of this complexity and nonlinearity, our research often takes a data-driven modeling approach. And that allows us to try to build in these more complex and often nonlinear mechanisms to better understand how environmental change is affecting infectious disease systems. So today I'm going to break the talk into two parts. The first, which will be the majority of the talk, will focus on the impacts of climate change on vector-borne disease. And then in the second part of the talk, I'll move into talking about impacts of land use change on vector-borne disease. So how will climate change affect vector-borne disease transmission? This is work that was led by a number of really great postdocs and grad students in my lab over the years. And it really started out with this question, is a warmer world going to be a sicker world? Around the early 2000s, this picture started to emerge in the literature that we might expect, especially tropical diseases like malaria, to start expanding because of climate warming. And this shows just one example of this type of research paradigm, which is a model projecting changes in the climate suitability for malaria transmission by mid-century. And what they're showing here is the predicted change in the number of infectious bites per person per year. The important thing to notice here is this scale is almost entirely positive, ranging from green, small increases, to red and purple, very large increases in climate suitability. We do see a little bit of declines in climate suitability along the Sahel, where it was projected to become too dry. But otherwise, this body of research, and that's not just this one paper, it's many papers suggesting that climate warming was expected to broadly expand the suitability for malaria transmission, representing a really big public health impact of climate change. But what we started thinking about in looking at this research is whether there might actually be a more complex and nuanced picture underlying this. And the reason for that is if you look at the vector-borne disease transmission cycle, it's a complex cycle. So for a mosquito-borne disease, you have female mosquitoes that lay their eggs on the side of a body of water. Those eggs then have to hatch, go through 4 larval stages, and then pupate, and then eventually emerge as adults. And each of those life history processes, from survival to fecundity to development, depends on the metabolism of the mosquito. The mosquito is a small-bodied ectotherm, cold-blooded insect. So its body temperature is matching the temperature of the environment around it. You may recall from your chemistry and biochemistry classes that reactions, like the reactions that underlie metabolic processes, can be temperature dependent, because as the temperature increases, reactions can speed up to the point that enzymes begin to denature and things fall apart again. So there's a lot of temperature sensitivity in these life history traits that are related to metabolic processes. Now, of course, the part of the mosquito's life cycle that they're most famous for is the fact that females bite vertebrate hosts to get a blood meal, and they use that blood meal to produce eggs. And that biting rate is also dependent on the metabolism of the mosquito, so therefore dependent on temperature. The parasite, for its part, has hijacked this mosquito life cycle, so that if that blood meal contains parasites, such as viruses or protozoans, instead of getting digested and excreted out of the mosquito's body, they're able to actually break through the mid-gut barrier, disseminate throughout the body, and eventually bind to the salivary glands, where they get injected into that next host that gets bitten. And this process is called the extrinsic incubation cycle. And both the rate of that process, as well as the probability of successfully becoming infectious, so it's called the vector confidence, those are dependent on the mosquito's metabolism and therefore dependent on temperature. So if we look across this vector transmission cycle, we can see lots of different places where temperature comes into play, suggesting that there may be a complex picture here of how temperature might affect mosquito-borne disease transmission. But beyond that, we have really strong expectations for how temperature actually affects these different life history processes, which I alluded to in describing the metabolic cycle. One of the most widely established patterns that we have in ectotherm physiology is the pattern of nonlinear thermal responses. So whether we're talking about the performance of development rate or survival or reproduction or biting or behavior, these processes tend to respond nonlinearly to temperature. So there's a critical thermal minimum below which the process cannot occur. And then as you exceed that minimum, the process increases up to an optimal temperature before it drops off, often really steeply up to a critical thermal maximum. So again, there's really widespread support for this nonlinear thermal response in the ecological literature. There's empirical evidence from thousands of different ectotherm taxa and traits showing these nonlinear thermal performance curves. And there's theoretical support for why we would expect this kind of nonlinear thermal response. And yet, when we look in the vector-borne disease literature, particularly those models projecting that a warmer world will be a sicker world, we often see this disconnect where thermal responses are assumed to be linear or monotonic, meaning they only go in one direction. So for example, we see the idea that temperature is just speeding up the parasite development cycle and not really considering the potential for that upper thermal limit to occur. So the goal of this research is to reconcile this disconnect between the vector-borne disease literature and ectotherm physiology literature, and to ask whether nonlinear thermal performance curves are the norm in vector-borne disease transmission. So more specifically, how does temperature affect vector-borne disease transmission? The research approach here is to go into the literature and look at studies where entomologists have held mosquitoes at different constant temperatures and measured these life history traits that underlie transmission, like biting rate, surveillance rate, pathogen development rate, and so on. And from these empirical data represented by the dots here, we can fit a continuous thermal performance curve to estimate how each trait responds to temperature. But then we want a way of synthesizing overall, how does the transmission process as a whole respond to temperature? So from there, we can build up a model of R0, or the rate of spread of a novel pathogen into a fully susceptible population, as a function of these traits that are temperature sensitive. So this is our expression we're using for R0. The key thing to note about this expression is that it's a nonlinear function of a bunch of mosquito and parasite traits that are themselves nonlinear functions of temperature that we've estimated from empirical data. So for our purposes, R0 is really kind of a relative measure of how suitable the temperature is for transmission, all else being equal. So it gives us a way of estimating the temperature suitability by incorporating the impacts of temperature across the mosquito life cycle. Then we want to take this mathematical model that's been parameterized with experimental lab data and compare it with field observations to ask whether we're actually reproducing patterns of transmission versus temperature that we see in the field. And finally, with that validated model, we can then use it to project both the current and future temperature suitability for vector-borne disease transmission. So we first applied this method to falciparum malaria transmission with Anopheles mosquitoes in the context of Africa back in 2013 when I was a graduate student. And what we found in this work was really strong support for nonlinear thermal performance curves. So this is one example, the mosquito development rate from egg to adult. You see this classic hump-shaped thermal response. And in fact, if we look across all the traits that drive malaria transmission, we see strong evidence for nonlinear responses across the board. So when we take these thermal performance curves and we plug them into our temperature-dependent R0 model, this is what we find. We predict that R0 peaks at 25 degrees and decreases to 0 around 17 or 18 degrees, and again around 34 degrees Celsius. For context, that's about 78 degrees Fahrenheit is that optimal temperature. Now, by contrast, when we looked in the previous literature, all of the previous malaria transmission models that had used this type of approach predicted an optimal temperature of 31 to 32 degrees Celsius. So our new estimate is 7 degrees cooler. And that really has big implications if we're thinking about the impacts of climate change, because, 25 degrees, if we're increasing temperature above there, we might be predicting a decrease in transmission as opposed to an increase. So this really highlighted for us the importance of validating these models against field data to see whether we were accurately reproducing patterns of transmission versus temperature. And what we found when we validated this model was strong support for this predicted 25-degree optimal temperature in field data. So what I'm showing in the circles here are the entomological inoculation rate, or the number of infectious bites per person per year, which was measured across Africa over a 3-decade period. And what you can see here is that there's quite a bit of variation. Not all of it is explained by temperature. A lot of it is not explained by temperature. But the highest rates of transmission were measured right around that predicted 25-degree optimal temperature. And we see strong evidence for decreases in transmission above 28 degrees Celsius or so. So providing pretty strong support for this new predicted optimal temperature. Now, we're also acknowledging that there's lots of variation not explained by temperature. So we binned these data points by temperature into groups of 10, and we're filling the maximum within each of those bins as a place where we would expect that other factors are least limiting on transmission, and therefore temperature may have the strongest impact. And so just as a way to visualize the effects of temperature, you can kind of look at these filled circles and see that those filled circles, those maximum transmission points, are also highest around 25 degrees Celsius.
Just
Sure.
a clarification,
Yeah.
this is for anopheles and specifically for a species of malaria?
Anopheles gambiae and falciparum malaria,
Falciparum,
yeah,
okay.
in Africa,
Thanks.
yeah. So then, this was work that was done a while ago, and the validation was based on entomological data, but we were then interested in whether this could actually predict human malaria burden. And since coming to Stanford, I had the opportunity to start working with Dr. Desiree Lebeau, who I know some of you may know. And Desiree's group has been doing a pediatric cohort study on malaria and arboviruses in Kenya, and working with data from over 2,000 children across 4 different villages in Kenya. They measured the incidence of malaria as smear positivity against the temperature during the transmission season. And that's what I'm showing in these triangles here. And in particular, what you see is that the highest rates of incidence are occurring around 25 degrees Celsius. And we see strong evidence for that declining incidence as we increase temperatures above 25 degrees Celsius. Again, supporting the idea that the thermal optimum for malaria transmission is 25 degrees Celsius. So after seeing this big disconnect in the malaria literature, which was arguably the best studied vector-borne disease with respect to temperature, we were then interested in applying this research approach to other vector-borne diseases, particularly mosquito-borne diseases. And we applied the same research approach. So falciparum was the first one that I just showed you. We also applied it to dengue, chikungunya, Zika, West Nile, Eastern equine encephalitis, Sinbis, Rift Valley fever, Ross River virus, basically every mosquito-borne pathogen that we could find data in the literature for. And what we found across the board was evidence for nonlinear thermal responses. So here again, I'm just showing you the data for one trait of one species, just to give you an example. This is the mosquito development rate of Culex annulirostris, which is an important vector species in Australia. But we see support for these nonlinear thermal responses across all the major vector species, including the EIDs vectors of dengue, the Culex vectors of West Nile, and the Anopheles vectors of malaria.
Yes, studies account for change in human behavior. I mean, if it's very hot, more people will spend time.
Yeah.
outdoors.
These are all just entomological studies in the lab. So at this point, we're only, I'll get to that part later, but at this point, we're only basically saying, what does the mosquito do in response to temperature?
And how much role does humidity, ambient humidity
Yeah.
play in this?
Humidity plays an important role, especially in survival. But what we've found is that humidity doesn't interact with temperature in the sense that it doesn't change the shape of the thermal performance curve. So we can now look across all these different life history traits and all of these different vector and parasite species, and we see the consistency of the nonlinear thermal response. So each of these panels is a different trait, the biting rate, fecundity, mosquito development rate, immature and adult survival, and the incubation rate of the parasite in the mosquito. And each of the lines is a different vector or vector and parasite combination. What you see if you look at these curves is consistently they have hump-shaped responses to temperature, as we would predict from ectotherm theory. But what we also see is that there's variation among different species and among different traits, suggesting that it's important to actually measure it for a given species and a given trait because there are differences. If we now put these together into our temperature-dependent R0 models for each of these systems, this is how we predict that they respond to temperature. So here I have temperature on the x-axis, I have relative R0 on the y-axis, so scale just so that they all peak at 1. We're really comparing kind of left to right, where are their thermal optima and where are their limits. And what we see here across these different systems is that consistently they respond non-linearly to temperature. They all have an intermediate optimal temperature, and that optimum varies across different systems, as well as the thermal minimum. There's quite a bit of variation there. There's a little bit less variation in the thermal maximum across systems. Just to highlight what some of these pathogens are, we see Zika and dengue transmitted by Aedes aegypti have some of the warmest thermal optima around 29 degrees Celsius. Malaria peaks right in the middle at 25 degrees Celsius. West Nile and Triple E peak at lower temperatures, around 23 and 24 degrees Celsius. And I'm not going to go through validation of each one of these models. We've attempted different things for different systems depending on what data are available, but I did want to show something kind of interesting, which is that our understanding of these nonlinear thermal responses of transmission helps to reconcile some previously contradictory findings in the field. So here I'm showing a review that a previous postdoc in my lab did, looking at just statistical studies of the impact of temperature on transmission of dengue in the field. And what she found is that these statistical effects of temperature on transmission varied from strongly negative to basically 0 to strongly positive. So if you just look at these studies, you would say, we don't really understand a lot about how temperature is affecting dengue transmission. But if we rearrange these effect sizes based on the average temperature at which the study was conducted, we see that our thermal performance curve helps us to understand why we see large positive effects of temperature at the part of the curve that's really accelerating. We see basically zero effects of temperature when we're already near the optimum. And we see negative effects of temperature when we get to temperatures that exceed the optimum. We wanted to further quantify this effect by doing a meta-analysis. So we went into the literature again, and we more systematically reviewed studies of dengue and temperature. And we wanted to test this prediction that we would see the largest effects of temperature. So we're basically thinking about the slope of R naught with respect to temperature. We expected to see the largest effects where this curve is the steepest, around 25 degrees Celsius. And we expected to see this effects declining towards zero at lower and higher temperatures. And what we found in this meta-analysis was support for that prediction. So the highest correlation between temperature and transmission in these studies was around 24, 25 degrees Celsius. And the effects of temperature on transmission declined to zero or even negative at both low and high temperatures.
Erin?
Yes.
When you're thinking temperatures, it's like, it's not maximum daily temperature. You're thinking like week long, you know, over what time
Yeah.
period, what metric of temperature.
One of the variables in this study that I didn't go into detail on was which temperature they used. And there is some variation, as you might expect. When they used minimum temperatures, there tended to be more positive effects. When they used median or average temperatures, there tended to be kind of medium level effects. And then when they used maximum temperatures, the effects tended to be more negative.
So from a sort of biochemical point of view, which temperature do you expect to be the most important, like over what averaging?
Yeah, I mean, they're all just summaries, and the mosquito is experiencing all of them. So the nonlinear averaging part of this is kind of it's like a little bit inside baseball, but it's you know, it's an interesting component. The temperature variation can have nonlinear effects as well. That there are ways to kind of integrate over these nonlinear thermal performance curves to incorporate the effects of temperature variation, which I think is important both at daily and seasonal and interannual scales. So it's important at all the scales. But that's one of the challenges with validating these models is that temperature is not constant in the field, inconveniently for us.
Okay.
But I want to now dive into something that is very brand new. This is literally the first time I've ever presented it, and I'm so excited to be presenting it today. All of this validation work that I've shown you so far is kind of correlative. Like, oh, if we plot temperature and we plot dengue, they kind of agree with what we would predict from a model. Here, for the first time, we're attempting to do a causal inference approach, looking at effects of temperature on dengue. So these are all sort of relative. What is the more suitable and less suitable temperature for dengue transmission? Here we're asking, can we actually quantify the impacts of temperature variation and temperature change on dengue burden or dengue incidence using a causal approach? So here I'm focusing on dengue to give you a tiny bit more background on this disease. Again, it causes 58 to 96 million cases per year. It's distributed throughout the tropics and subtropics in Latin America, Southeast Asia, and South Asia, as well as sub-Saharan Africa, although that burden is often under-recognized. And it's primarily transmitted by two mosquito vectors, Aedes aegypti and Aedes albopictus. Aedes aegypti is the main vector in Latin America and Africa, and albopictus becomes more common in parts of Southeast Asia. The questions we want to ask here are, how does temperature affect dengue incidence quantitatively? And how do historical and future climate change impact dengue burden? So can we actually attribute past impacts of climate change, and can we predict what those future impacts will be? To do this, we're going to use what's called an econometric panel regression approach. And the goal of this approach is you have units such as districts or counties or states, and you're comparing them to themselves over time. So you're asking, you know, controlling for whatever might be idiosyncratic and unique about this particular unit. If this particular unit has higher than average temperature, does it also have higher than average dengue, while controlling for other potentially confounding variables? And that's what makes this a causal analysis. And then we want to test for specific hypothesized effects. So in this case, we're focused on temperature. So in contrast to a lot of kind of biological modeling, where you might be looking for the best possible model to explain the most variation in the data, that's not the goal here. The goal is to try to test this hypothesized effect of temperature while controlling for as much possible confounding variation as possible. So this is the structure of this type of model. We have dengue incidence as our response. We have temperature as our predictor, and in this case, we're using ERA5 temperature data. We're using dengue incidence from 22 countries, so over 7,000 unique units within those 22 countries. an average of about 11 years. Some of the time series range from about 10 years to 30 years or so. And we're including a polynomial function of temperature, so something that can have a flexible shape because we do expect that nonlinear response to temperature. We wanted to control for precipitation because, of course, that's expected to affect a mosquito-borne disease as well. So we include data on precipitation for each unit. And then we include these fixed effects, and that's really the key part of these panel regression models. The fixed effects control for the unit, so they subtract the average for a given county or a given state. They also control for the country year variation. So, for example, in Peru, was this a particularly bad year for dengue? And they control for country month variation, which is kind of capturing the seasonality processes that might vary country to country. So what's left behind is this kind of small amount of variation in temperature and small amount of variation in dengue. And we're really comparing those two to each other to ask what that relationship is. And then this is a population weighted Poisson regression. So we're weighting these units by population, so their effects are more informed by the higher population size locations. This is what our data set looks like. So this is a map that's showing temperature variation in the red colors and dengue variation in the blue colors. So the colors that look sort of purpley black are places with high temperature and high dengue burden. You can see that our data set includes many of the countries, most of the countries in Southeast Asia, and most of the countries in Central and South America, as well as part of Mexico and some of the Caribbean. There's some notable data gaps as well, which I'll get to later. And these are just some examples of what some of these time series look like, summarized at the country level, although the analysis is done at the smaller spatial scale. So getting to the exciting part, this is what we found in this regression. This is our polynomial response to temperature that we estimated. So temperature is on the x-axis, and the relative log of dengue incidence is on the y-axis. And you can see that this peaks around 28 degrees Celsius or so. In the bars, we're just showing the average temperature across the countries in the data set, highlighting Bolivia being the coolest temperature in the data set, Mexico, Brazil, Indonesia, and I can't remember what KHM stands for, as being the warmest temperature, Cambodia, there it is. So this is our estimated functional relationship from our panel regression model. We can compare that with our temperature dependent R0 model for Aedes aegypti and Aedes albopictus. So recall that Aedes aegypti R0 peaks at 29 degrees Celsius. For Aedes albopictus, it's about 26 degrees Celsius. If we compare these predicted thermal optima, 26 and 29, with our estimated response, we see that our peak at 27.8 is kind of pretty much in the middle of those two, like about what we would expect given the transmission by these two vector species. We can also not just focus on the optimal temperature, but look at the places where temperature is most impacting transmission. So that's where this line is the steepest. And in this case, that's around the temperatures of Bolivia and Mexico, like centered around 20 degrees Celsius or so. And if we kind of look at where that falls on the temperature dependent R0 curve, we can see that this is a place where temperature dependent R0 is increasing pretty steeply. So what I'm trying to show here is that there's pretty good agreement between these really different approaches of estimating the effects of temperature on dengue transmission. One is inferring from a global data set of observed dengue transmission, and one is inferring from a mosquito entomological data set from experiments. What we find in this work is that warming has already expanded dengue transmission. So these are the different countries in our data sets, and the little hatch marks are the different units within countries. They're ordered by the average temperature in each country. So the places where warming has had the biggest impacts already are the countries that have cooler average temperatures. So those are places like Bolivia, Peru, Mexico, and Colombia. These are places that have, large mountain ranges that kind of moderate the temperatures. And in these places, dengue has already, about 40% of the existing dengue burden can be attributed to climate change that has already happened based on counterfactual historical climate scenarios, saying what would have happened in the absence of anthropogenic forcing on the climate. If we look at that on the map, this is the percent of dengue that's already happened due to climate change that's already happened. And again, you can see that that's kind of concentrated around the highland regions of South America and Central America, as well as to some degree in Southeast Asia, although you can see there's a lot more blue colors where climate change is not having as much of an impact because those places are already on the warmer side. We can also plot these historical impacts of climate change on dengue as a function of the current incidence. And you can see, and then this is the size of these points is based on population size. So you can see a few of the larger countries here, like Mexico, Brazil, Indonesia, you can see that they've had between 20 and 40% of dengue burden that's already existing is due to climate change that has already occurred. So this is suggesting that we've already seen very large impacts of climate warming on dengue transmission. What's going to happen going forward with future climate warming? Here we're showing the projected response by mid-century under the worst case climate scenario, the currently accepted worst case climate scenario of SSP 3-7.0. And what we're expecting is an exacerbation of these changes. So the places that already have experienced a lot of warming driven dengue are expected to experience even more, particularly those highland regions of Latin America and parts of northern Vietnam and Laos. Again, we can show that by country. And again, we can see that in some places we're expecting a doubling or even more of dengue burden by mid-century because of climate warming, because the climate is driving temperatures even more into that suitable range for dengue transmission. And that's really, here we're showing the derivative of the response of dengue to temperature, showing where is that response the steepest. And that response is the steepest right around these kind of 18 to 20 degree temperature ranges, which is right where parts of Bolivia and Mexico and parts of Brazil are. So we're expecting to see expansions in places like southern Brazil as well. Now, places like Indonesia that already have very warm temperatures are not necessarily expected to get much worse because of climate change, because their temperatures are already highly suitable. Either.
Is this accounting for where the people are?
Yes.
Okay.
Yeah. So these municipalities are weighted by population size. Okay.
With any of that piece, following that.
Yeah.
Population expansion, those are some of the biggest areas of population expansion in the world.
Yeah.
Population density is a potential factor in increase in dengue transmission.
Yeah, it definitely is. Right now, that's not built into this model at all. So that would be kind of on top of the temperature mediated changes. Yeah. David.
You said you were going to explain why some of the countries don't have any data. I mean, it looks like Cuba, Ecuador. I'm not sure what the other one was, French Guiana, maybe.
Yeah.
Cuba's been really notorious for not reporting.
Yeah. Yeah.
And Ecuador too.
Yeah. Well, okay. So this gets at one of the drawbacks to this approach, was it's really data needy. So what you need is repeated reports within at smaller than country units, like admin 1 or admin 2 units over time. So you need a place that has been, repeatedly reporting at least 5 years or so, multiple within country units. And we dug as hard as we could for data in all of these countries, and a lot of them we just couldn't find. So they may have data, at the country scale or going back, but not going back very far, but we couldn't find those kind of consistent case reports that we needed for this particular approach. So, and some of the biggest gaps are actually all of sub-Saharan Africa and India. India has a huge dengue burden, and we know that the data exists for India. We could not get them. So if anybody knows someone in, in this Indian Ministry of Health that can help us find these data, we would love to include India. We think in sub-Saharan Africa, the data just probably aren't being collected systematically enough at this point, but I'm going to get to in a second what we think might happen in other places as well, because we can project just based on temperature. We just can't do this kind of causal inference approach without this kind of data.
I might be able to help with India.
Oh, awesome.
Talk
Great.
about that.
Okay. So just comparing under different future climate scenarios, like could it be better or worse depending on the climate scenarios? Yeah. This is SFP 1-2.6. This is kind of considered the best case scenario. Like if we meet the Paris Agreement goals, this is what climate warming would look like. We can see that there's still going to be a projected doubling of dengue risk in some countries, but it's much worse under the worst climate scenario. So, sorry, each of these points is a country. And these error bars, so it's summarized at the country level here, and the error bars represent both the uncertainty in the model and the uncertainty in the climate projections. So you can see that there's some wide error bars, but we're still pretty confident these are going to be large increases due to temperature warming. If we compare the best case and worst case climate scenario, this is now the difference between those two. You can see that there's greater than 30% worse in the worst case climate scenario. So it's definitely worth trying to mitigate climate emissions as best we can. But even under the climate change we're projected to already have, dengue is really expected to expand. So getting to those key data gaps now, you can see our map just conveniently is centered on the Pacific Ocean and is missing like most of the world where there could be dengue. Maybe there already is dengue, but we don't have really good case reports. So we're missing all of sub-Saharan Africa, where we think dengue is an under-recognized burden now. We're missing India. We're also missing places where dengue could be having a range expansion, where there's not a lot of dengue now, and there hasn't been enough historical dengue to have consistent case reporting, but we could see range expansions. We don't really have good systematic information on control measures, so this kind of, some of that is absorbed in the fixed effects, not to get too technical about it, but it would be great if we had better information about what control measures are in place. And we're not really dividing this out by vector species right now. So we're just kind of assuming this is all one system. And yet, this approach is actually very conservative because it assumes that none of the differences between countries or the year-to-year differences within countries or the seasonal differences within countries are driven by temperature. So this is just that kind of additional anomalous variation that we can really causally attribute that temperature variation to dengue. So we could do this in less conservative ways. I see that I'm starting to run low on time, so I might skip a few slides here. Very interestingly, a group just did a very similar approach, almost the exact same thing, but with malaria in sub-Saharan Africa from 1900 to 2015. Just to show you my favorite part of this work, this is their inferred effective temperature on malaria transmission on the bottom in the pink curve, and this is our predicted temperature dependent R naught on the top in the black curve. And you can see that they have basically the exact same thermal optimum of 25 degrees. So again, providing an independent validation of this 25-degree optimal temperature for malaria transmission. I'm going to not spend time going through their results, but it's cool. So what we find is that causal inference validates our mechanistic predictions of how vector-borne disease responds to temperature. Dengue peaks at higher temperatures than malaria. Historical climate change has already expanded dengue transmission while it has shifted malaria transmission. So that was the key result of that paper I skipped through, showing that we've seen a shift in malaria transmission from more of kind of Central and West Africa into Southern Africa and Highland Africa. And both of these changes are expected to be exacerbated with future climate change. And these approaches are conservative and very data limited because they do really require a lot of very rich data to be able to do these methods. So we can go ahead and just project the temperature suitability without kind of doing any causal inference and ask, how suitable is the temperature for these diseases, all else being equal? How will climate change affect the spread of these diseases? Well, if we're starting at these kind of cooler than optimal temperatures, then of course, any temperature warming is expected to drive up basically all of these diseases. If we're starting at the warmer than optimal range, then any further warming is only expected to drive down these diseases. And there's a really interesting range of temperatures between about 23 and 29 degrees Celsius, where we're seeing differing responses across different systems. So we could see the rise and fall of different pathogens with climate warming. And just to show you this again for malaria and dengue, now we're just projecting how the temperature suitability for these two pathogens will change in sub-Saharan Africa, because that was one of the key data gaps for dengue in particular. This is how the climate suitability, the temperature suitability for malaria transmission overlaid with people. So the number of person months of high temperature suitability per year in sub-Saharan Africa is currently centered on coastal West Africa and Central Africa, but we expect that hotspot for transmission to kind of contract and shift towards high elevation East Africa and Southern Africa with climate warming. By contrast, for dengue, which we know is present and very widespread in sub-Saharan Africa, but often underdiagnosed, we have a similar current hotspot of dengue suitability, but we expect that hotspot to broadly expand throughout sub-Saharan Africa with warming, including into some of those same regions that are expected to have expanded malaria transmission. So this is a really important message from a public health perspective, because although they're both mosquito-borne diseases, the way that we control them is completely different. The diagnostic tools that we have for malaria and the therapeutics that we have for malaria do not work on dengue. Even the vector control tools are different because the Anopheles mosquito is more of a night-biting mosquito, where an insecticide-treated bed net can be a very powerful tool. The Aedes aegypti mosquito is a day-biting mosquito that breeds in containers and has different ecology and behavior. So the types of vector control tools that we'll need to address this growing arbovirus dengue transmission problem in sub-Saharan Africa will be different. So to summarize the effects of temperature on vector-borne disease transmission, first, we find that the trait responses are nonlinear. So the traits that underlie the transmission processes. And it's very important to incorporate these nonlinear trait responses into our models if we want to accurately predict how temperature is affecting transmission. We find that the thermal optima and limits vary both across different vector and pathogen species and across different traits. And we find that climate change is often expected to drive shifts in suitability rather than widespread expansion. Although for dengue, we may see more widespread expansions. And dengue has been expanding while malaria is shifting because of their differences in thermal optimum. Now, all of this is mediated by human activities. So this is an important caveat and something that I want to dive into for the second part of the talk, hopefully a little bit more quickly than the first part. So here we're going to address the impacts of land use change on vector-borne disease transmission. And this is work that's been spearheaded by 3 postdocs and an undergrad in my lab who've done some really fantastic work, really inspired by starting to think about deforestation in the Amazon. This is what deforestation in the Amazon looks like. It's this kind of fishbone pattern where you get a road that provides access deep into the Amazon and these little kind of offshoot roads. You get this really extensive deforestation that occurs right at the interface with the forest. So it's almost this fractal-like pattern of creating forest edge where people are living and working, mostly subsistence farming, at these forest edges. And in the Amazon, malaria is transmitted by a forest edge-associated mosquito called Anopheles darlingi. And it's caused primarily by 2 parasites, Plasmodium vivax and Plasmodium falciparum. Now, this work has been published for several years, so I'm going to just skip going over the methods altogether. I'll give you the citation if you want to see the methods, and I'm happy to talk about it in more detail. But in short, it uses a lot of those same causal inference panel regression models from econometrics that I just showed you. What we found in this work is so again, it's an econometric causal inference approach. We found evidence for a bidirectional feedback between deforestation and malaria, where deforestation promotes malaria when it's within the forest. So when it's a situation where deforestation is creating forest edge and bringing people in contact with the forest edge, we see that deforestation promotes malaria with about 6.4 additional malaria cases per square kilometer of forest loss. By contrast, malaria incidence actually reduces deforestation. And we can estimate this effect at about 0.07 fewer square kilometers of forest cleared for every case of malaria. We think this is through some sort of socioeconomic mechanism where either people are too sick to work their land as subsistence farmers or they're abandoning settlements. But we're very curious as to what this mechanism is more specifically. And we find that this deforestation and malaria feedback in both directions is strongest in the frontier regions of the Amazon at the earliest stages of forest clearing, where you really get that kind of forest edge frontier malaria phenomenon. Whereas if you move into the outer Amazon, where most of the forest is already cleared, these effects are much weaker. And this has important policy implications because, as you probably know, the previous administration in Brazil was really aggressively promoting clearing of the Amazon and development in the Amazon, quote unquote development. And we saw a massive increase in deforestation in places like the Jamanchim National Forest. So here you can see that fishbone pattern really clearly from aerial imagery. Deforestation in 2021 surged to over 13,000 square kilometers. What this type of modeling approach allows us to do is to quantify a potential human health impact of that deforestation. So just using our model that we inferred from the previous data, we would expect about 83,000 additional malaria cases because of that deforestation. Now, in 2022, Brazil adopted a goal of eliminating malaria transmission completely by 2035. And by 2025, their goal is to get down to fewer than 65,000 cases per year. We can see that that's going to be impossible unless they eliminate this deforestation. So it really, again, helps us to quantify how environmental policy can influence human health and how human health policy has to take into account these environmental drivers and the socioeconomic drivers that are underlying this. So this work really inspired us to start thinking more broadly about how different types of land use change might promote different vector-borne diseases. And we kind of came up with this hypothesis that there could be distinct land use niches for vector-borne disease transmission. So for the malaria system I just showed you, we're in this kind of frontier clearing and subsistence agriculture stage of land use transition. So we start out with natural ecosystems that are intact. There's frontier clearings and subsistence agriculture, then there's more intensified agriculture, and finally urbanization. So malaria in the Amazon is in this kind of frontier subsistence stage. But we could kind of, based on our knowledge of the vector and reservoir host ecology of different diseases, we could hypothesize how different diseases would map onto this land use gradient. So we might expect the more sylvatic diseases like yellow fever and cutaneous leishmaniasis that have wildlife reservoir hosts and forest-associated vectors. They might peak at these very earliest kind of natural ecosystem stages. Malaria is peaking kind of in the middle. Visceral leishmaniasis, we think, is kind of a peri-urban / agricultural disease. And then Zika, dengue, and chikungunya transmitted by the urban vector Aedes aegypti, we would expect to peak kind of at the latest stages. So we wanted to test this hypothesis and see if we could actually quantify land use niches for different vector-borne diseases. So to do that, we turned to Brazil, again, as we did in the malaria work, because Brazil is a very large and very diverse country that has a large number of different ecosystems and climate zones, a large number of people, a large number of cultures, and a large number of vector-borne diseases. So here I'm just showing maps of the average incidence of these diseases in the last decade or so for dengue, chikungunya, Zika, malaria, cutaneous leishmaniasis, and visceral leishmaniasis. So these are important vector-borne diseases in Brazil that are widely reported. We wanted to ask whether we could identify a land use niche for transmission of each of these diseases. So how do we actually quantify this land use gradient, which we know is really complex and multidimensional? How do we kind of collapse this down into something that can be measured on a single axis? In the last 5 years or so, there have been advances in the science of human pressure mapping, where people have developed indices called the Human Footprint Index, which is basically a multidimensional index of how much human pressure is occurring in a given location. And it can be measured using satellite imagery. So specifically, this quantifies built environments, energy and transportation infrastructure, so roads and lights, agricultural lands, and human population density. It multiplies it all together in a weighted average to give you something that ranges from 0 to 50. And this is what human footprint looks like globally in 2009, but this has been calculated now over several time points globally. So we now have this index that allows us to estimate human footprint over time and over space. And we were curious whether this human footprint index would be predictive of these different vector-borne diseases in Brazil. So how do vector-borne diseases with different ecologies respond to human footprint? Here we're using a machine learning approach as opposed to our causal inference approach, because we're really more in the hypothesis generation stage here. We don't really have a specific causal hypothesis to test yet. So we're asking for each of these 6 diseases, is their occurrence predicted by human footprint, population size, climate variables, and land cover variables? And specifically, is human footprint an important predictor of each of these diseases? So this is a map of what each of these variables, just some examples of what these variables look like. Human footprint across Brazil, population size, temperature, and forest cover. Again, we're focused on human footprint here. And what we found with this machine learning approach was that for each of these 6 diseases, human footprint was among the top predictors of its occurrence in a given municipality in a given year. And if we look at that functional relationship that the model inferred, this is human footprint ranging from 0 to 50 on the x-axis. And this is a rescaled estimate of the probability of occurrence for each disease on the y-axis. We see these strong threshold responses. So some diseases start out highly likely to occur at low human footprint, but then they kind of tank. As you increase human footprint past a given threshold, suddenly the occurrence of these 3 diseases, malaria and both forms of leishmaniasis, become a lot less likely. By contrast, these 3 diseases that are transmitted by Aedes aegypti, the urban vector, really increase with human footprint. We don't see them in the more intact environments, and we start to see them as we exceed this threshold of human footprint. So let's dive a little closer into what these different values of human footprint actually mean. We know that something less than 4 is basically an intact ecosystem. There's minimal biodiversity threat, there's minimal human impact. As we move from 4 to 7, that's kind of agriculture. And then above 12 is intense human pressure, so basically an urban area. And between 7 and 12 is that intensifying agriculture and transition from peri-urban into urban areas. So we can now use this kind of mapping to identify the land use niches for these vector-borne diseases. And what we find is that malaria has this kind of intact ecosystem to early stages of agriculture niche. Once we hit a human footprint of 5, malaria incidence is really much less likely to occur. Visceral and cutaneous leishmaniasis kind of persists through the agricultural intensification stage to a human footprint of about 10 to 11. Dengue starts to pick up around that same level. So as we're starting to see the decline in leishmaniasis, we start to see increases in dengue. And then chikungunya and Zika had even higher levels of threshold values of human footprint. Now, the final thing I want to show you really quickly is thinking a little bit more about what's underlying the ecology, particularly of leishmaniasis, visceral and cutaneous leishmaniasis, because these are a zoonotic vector-borne disease system. There's actually several parasites in the Leishmania genus. This is the parasitic life cycle for leishmaniasis. So it has these parasites that are transmitted by sandfly vectors that are then going through some sort of reservoir host. And then they're going from the reservoir host back to the vector. Humans get infected by leishmaniasis through spillover. So humans are typically a dead end host for leishmaniasis. There are dozens of known reservoir and vector species, but we also expect that there are many more that are yet unknown. So we were interested here in trying to say, based on what we know about the known vectors and hosts, what are the most likely unrecognized vectors and hosts? So here again, we applied a machine learning approach where we built in information about morphology, life history, biogeography, and phylogeny of these known vectors and hosts. And said, based on this, if we look at the broader set of sandfly species and vertebrate species in these systems, which species are most likely to be hosts and vectors? What we found for the hosts is 136 newly predicted host species, primarily including rodents and bats, but also including things like non-human primates and sloths and armadillos. The key ecological predictors of those hosts were species that occur around high human density, high crop cover, high urban cover. They have fast-paced life history, and they are associated with warm temperatures. So these are kind of human-associated species. For the vectors, we identified 13 newly predicted vector species, and these tended to be synanthropic, so associated with human environments, but also associated with high forest canopy, low human modification, and a broad range of ecotones. So these are vector species that are really thriving in the kind of beginning stages of human impacts, where humans are present, but there's still a lot of kind of natural space present. So if we look at the land use predictors, we can see that for both the reservoir hosts and the vectors, there are key land use variables that might explain their association with human footprint. So for the reservoir hosts, they tend to be associated with crop cover and urban cover, whereas for the vectors, they tend to be associated with high forest canopy and low human modification. And together, when you get the intersection of those types of habitats, you're thinking about sort of a fragmented edge habitat where you have crop cover, urban cover, but also intact forest nearby. And that's where we would expect the nexus of leishmaniasis transmission to be occurring. So to summarize the effects of land use change on vector-borne disease transmission, we do find evidence for distinct land use niches for different vector-borne diseases. We also see sharp threshold effects, which might otherwise seem unexpected. So over a fairly small transition in land use and human pressure, you see the sudden emergence or disappearance of a vector-borne disease. And this is driven by shifts in the host and vector community that either promote or disfavor a given vector or host. There's also feedbacks with human behavior here. So humans actually can respond to the disease burden and change their behavior. And all of this is interacting with the effects of climate change. So to conclude, how will global change affect the spread of vector-borne diseases? We see evidence for the rise and fall of different vector-borne diseases across key thresholds. So for example, key temperature thresholds ranging from about 18 to 25 degrees Celsius, and key human footprint thresholds ranging from about 5 to 12, or that kind of early stages of agriculture into peri-urban zone. And more generally, we can predict and we can even quantify the impacts of human impacts on the environment, on human health. And it's important to use these data-driven modeling approaches so that we can better inform how environmental policy feeds back to affect human health and vice versa. So thank you very much, and I'm happy to take any questions.
Question about the first half of your talk. When you're predicting the burden of these diseases over the temperature, I have been trying to do something similar for known vector-borne diseases, infectious diseases. And one of the criticisms that we always get is how to incorporate the population level immunity coming from multiple exposures. So if you see increase in transmission, of course, people are exposed multiple times throughout their lifetime, and that means they are getting their immunity is getting boosted from these multiple exposures. So I wonder how you. I actually don't know. I don't work on the vector-borne disease, so I don't know how much immunity plays a role. But I wonder how you think about that.
Yeah, it definitely does. That's a great question. There's like the layer of just temperature. What does temperature do to the transmission potential? Which for a vector-borne disease, it's easy to kind of separate out from the human component. But then there's the layer of how susceptible are people, and how many cases do you detect is going to depend on how many symptomatic cases there are, which depends on immunity. So in the panel regression models that I showed, the nice thing about those models is that a lot of these factors are incorporated into the fixed effects. So like the country year variation, for example, it's a fixed effect. It's like a dummy variable. You're like subtracting off the average for that country and year, which probably a lot of that variation is driven by variation over time and immunity. So like, you know, for a given, let's say for dengue, for a given country, they may have had a really bad dengue outbreak last year. And so population susceptibility is lower. So that kind of starts you off at a lower baseline, but then you can still kind of see the effects of temperature from that lower baseline. So that's where this kind of methodology can be useful, using fixed effects to kind of control for that potentially confounding variation that you can't really directly measure, but you can indirectly measure it. But another approach is to use dynamic SIR type models, like more mechanistic models where you model long-term dynamics. But then it's hard to really capture the endemic dynamics, you know, accurately. You have to get a model that can somehow reproduce patterns of transmission and patterns of immunity over time. So we've done that kind of work as well. It's hard to get models to actually match the dynamics of immunity and cases over time, but that would be an alternative approach. And then a third approach that we've applied actually in working with Ethan is this empirical dynamic modeling approach, which uses time series, so you can match time series up together, like time series of cases, time series of temperature, time series of rainfall. And some of our collaborators figured out a clever way to estimated population immunity based on recent histories of cases, and put those time series together. You can reconstruct this dynamical attractor and then use features of that dynamical attractor to infer causal relationships between them. It's a bit technical.
Yeah.
I'm not, yeah, but in that way you can say, like, given the level of human immunity that we infer is happening, what is the relationship with temperature? And in particular in this work on dengue that Ethan and I have worked on, we showed that you see these nonlinear effects of temperature on dengue transmission, but only when human susceptibility is at a high enough level. When human susceptibility is low, you don't see any effects of temperature because basically, like, the population is not susceptible enough to sustain a large epidemic. So there's multiple tools. I think it depends on what kind of information you have about the system at hand. Like if you have long time series, then these panel regression methods and these EDM methods can be really valuable. Otherwise, you can model the suitability of temperature, all else being equal, and, you know, not consider the impact of immunity at all.
Yeah.
Yeah.
I have a question. For areas where you have multiple vectors coexisting, multiple mosquitoes,
Yeah.
do you find that there's like a survival competition, considered an exhibition? You know, you don't often see people with dengue and like who be really bad luck.
Yeah.
So-
Dengue and malaria.
Right. So there's a
Yeah.
competition at the human level with what
Yeah.
is propagated, but is there a competition that see or survival benefit of some mosquitoes over others,
Yeah.
particularly with climate change?
That's a great question. The ones that come to mind are Aedes aegypti and Aedes albopictus. They're kind of like sister species that are both vectors of dengue, chikungunya, and Zika. And when they co-occur in the same place, Aedes albopictus tends to win through really interesting mechanisms. It can outcompete Aedes aegypti, but it can also cross-fertilize and cause Aedes aegypti to become sterile. So locally, you get Aedes albopictus often winning, and it's a less competent vector, but it's still a vector. So it's a little unclear. It's actually a question that we're really interested in and trying to write a grant about that right now to better understand the dynamics when Aedes aegypti and albopictus co-occur, or at least kind of occur close by each other. How does that impact transmission? And also within Aedes aegypti, it can transmit dengue, Zika, chikungunya, yellow fever. You tend to not see them co-circulating.
Yeah.
But that could be based on diagnostics and cross-reactivity. Like, it's a little hard to know if that's a real pattern or if it's just that we're not good at detecting co-circulation of those. Again, I think you'd have to be pretty unlucky to get multiple arboviruses at the same time. And the mosquitoes don't live that long, so the chances that they feed on someone that has dengue and someone else that has chikungunya at the same time, like, it's a little hard to know what's going on. I don't think mechanistically we have good estimates of how the viruses affect each other's confidence, or, you know, affect each other within the mosquito. I think a lot of times different vectors have unique niches within the environment. Like if you look at the Anopheles gambii species complex versus Anopheles stevensii, like Anopheles gambii is the traditional malaria vector in Africa, and it tends to be more rural, and it tends to breed in natural pools of water and kind of rural environments. And then you have Anopheles stevensii invading, which is a more urban vector that can breed in containers, and it acts a lot more like Aedes aegypti, and it actually can co-occur with Aedes aegypti, and there's competition with it. So I think to some degree the vectors are offset in their niches, but then to some degree they actually do interact and compete. So I think it's a really interesting frontier of this work, is like to investigate the community ecology of the vectors, and when do different vectors win, and how does that change the transmission landscape. But I don't know of any studies that have done that at a like in a field context,
The New York Times has been highlighting this recently.
Yeah, I know,
Enough in Stevens eye.
yeah,
Recent. Sort of series
Terrifying,
malaria.
yeah,
just
really
so
bad.
Well done. I have a very different question, and this may be difficult, but I'm just wondering if there's a way to try and put together sort of the land use changes, and I'm thinking of the Amazon basin in particular, and then environmental conditions that try and predict spillover events. Which, I think it's difficult because you have to have an idea of what's going to spill over.
Yeah, yeah.
You need to know a little bit about what's circulating in
Yeah.
the jungle, and in
Yeah.
animals and so forth.
Is that-
And then what's the combination of conditions that's most likely to lead to something new emerging?
yeah.
That's what our NSF grant, Ecological is doing our. NSF
Oh, cool.
grant.
There's the known unknowns and the unknown unknown. It's hard to predict the unknown unknowns, like what unknown viruses or pathogens are out there that we haven't detected yet. That could be the next SARS-CoV-2 or whatever. But there's the known unknowns, which are like yellow fever and leishmaniasis and Mayaro virus and, or Mapuche, exactly. Like some of these viruses that we know about, we know who the vectors are, we know what some of the reservoir hosts are. We've actually attempted this with yellow fever in Brazil, where we built up a mechanistic model of spillover, really inspired by Raina Plowright's work. I don't know if you know her, but she's at Cornell and you should totally invite her for a seminar. She's amazing. She's changed the way I think about disease ecology through this idea that spillover happens because the alignment of multiple processes that have to occur in the same place and time. So you have to have a competent vector. You have to have reservoir hosts. The reservoir hosts have to be circulating infection. The vector has to be biting the reservoir host. The temperature has to be suitable for the vector to become infectious. The vector then has to be biting people. The people have to be susceptible. And then you have to have a diagnostic process that allows you to detect the pathogen. So when all those things happen, that's when you see spillover. And you can individually measure and model those things as a function of land use and climate and stuff. So we built up an environmental risk model of spillover for yellow fever, and found that it actually did remarkably well. Like we could actually model by building up this process, this kind of process-based model, we could predict the probability of spillover of yellow fever in Brazil, where there's a good diagnostic system and it's a well-documented disease, with pretty high accuracy, like around 0.8 area under the receiving operating curve, to be kind of technical about it. And we then compared it to a machine learning model where you don't build in that ecological knowledge. You just say, here's a bunch of variables. Let's see if we can predict it. And we threw in our mechanistic predictor as well. And our mechanistic ecological model of suitability was the top factor in the machine learning model. So that kind of helped us to understand that, like, yeah, this actually is capturing some biologically relevant variation. One of the interesting things is we tried to build in human vaccine coverage as well, thinking, okay, places where there's low vaccine coverage, that's where we should see lots of spillover. But it actually went in the opposite direction. Places where there was high vaccine coverage, there was more spillover because they vaccinate in places that have a lot of yellow fever risk. So it kind of let us know that that was responding to the problem instead of causing the problem. And even places with high yellow fever coverage, vaccine coverage, still have enough gaps that there are people susceptible that are getting infected. So I think that type of modeling approach is really promising for pathogens where we know something about their ecology, like we know what the vectors and hosts are, and we can build up those models. For the unknown unknowns, people have been trying to build up models based on land use interfaces and really thinking about the interface between forests and wild habitats and people and where is their interaction between them and high population density. But I think it's harder because it is going to depend on the specific ecology of whichever pathogen it happens to be.
We should probably stop
Thank you
here.
for sticking it out,
Thank you
everyone.
very much.
Yeah, thanks for coming.
Good. Thank you.